in that particular source category, so you would have a low rate of misclassification, and you would have a low false positive rate of assignment to that particular source category. And then you can calculate all of these rates up and determine an overall rate of specificity for the library. Sensitivity, on the other hand, denotes the frequency at which the frequency at which known positive isolates that come from a particular source do get classified into that particular category, and also then how many samples, for example, contain these isolates that contain isolates that would be classified into that category. So if you have high sensitivity, then you have a very low false negative rate for your library. And so basically then we're looking for libraries that have high specificity rates and high sensitivity rates. So that's a really crucial parameter. And if you haven't seen the type of data that gets generated with, for example, an antibiotic resistance analysis library, this is some data that I had on hand, showing, so these are all dairy cow isolates, and you see that there's a battery of antibiotics that are employed to generate the antibiotic resistance pattern. And then you have, so this is what we would do is record the highest level at which the organism would grow on the antibiotic. And so if you read across, then that would form the antibiotic resistance pattern for a particular isolate. And we used discriminant analysis to do the statistical analysis and establish the classification rule for a particular library. And I'll show you some data in a minute from some of the work that we did. And what one would hope, and what the original concept was it would be that you could come up with a pie chart like this, where so you would have a certain percentage that came from swine and from cattle and from, here's coyote, sheep, I think there's feline, rats in here. So in other words, you could really, in theory, and the hope was that you could really finally parse out what percentage of the E. coli isolates, for example, in this water body came from all of these different sources. And again, let me reiterate that that was the theory. But I will show you some data quickly from a paper that we did, published in, was it 2000? I think it was 2000, yeah. And in this case, we used a library, an antibiotic resistance analysis library, and we utilized both fecal coliforms and enterococci. So we had two separate libraries, and we were really trying to compare, well, which one is more useful in this Florida watershed for categorizing the correct sources of these, or the sources of these bacteria. And so what you see in this table 4, which comes directly from the paper, and again, here's some isolates growing on plates to give you the idea of what the patterns look like, or the data that we use to generate the patterns. So we have here results from our fecal streptococcus library and our fecal coliform library. And this was a really kind of a cool test of the efficacy of the method, and it came about serendipitously. My husband happens to work for the DOH, and he called me one day and he said, oh man, there is a septic system drain field blowing out at restaurant X on road Y, and it is going into the ditch. And I was like, oh, I'm all about that. So off I go. Off I go with my test tubes and my gloves. And I get there, and I was not disappointed because that puppy was blowing out and right into the ditch. So it was great. And I was like, looking at all these people, like they're eating their McBurgers around the, like, ooh, nasty. But at any rate, it ended up to be a really nice test of the method, because what I did was I collected samples directly from the blowout area, so it would have been basically human sewage that's coming right out of that septic system, and then I collected samples at a nearby ditch, and then there was also a cow pasture that was downslope of that particular drain field. So it had been raining as well. I was really lucky. So I kind of got this dilution factor in the ditch, and this cow field over here. And I was like, oh, this is perfect. So analyzed the bacteria that we isolated from these samples and compared them to our library. And at the time, our library, you can see the sources that were in our library. We have human, bird, chicken, cow, dog, pig, and raccoon. And so here's our samples, the curb sample right next to the blowout, the ditch sample, the pasture sample, and then this would be the total isolates. And so again, fecal streptococcus and fecal coliform. And so what you see, here's the percentage of what was classified into the various categories. And you see that most of these isolates were classified into the human category in all of these at all of these areas. So obviously then the library was doing its job in terms of being able to detect where these isolates were coming from, what the source was. You see there's a few bird here, you know, it's about like 10% there and 6% there, and a few were classified as dog and raccoon. Here's a 7% and a 6%. But overall, the vast majority, you know, somewhere between 67 and 100% of all these isolates at the various spots were classified as human.
So
And
on the chicken, on that first one, would that be noise?
yes.
That would be
Yeah, there was no reason to think there was chickens around there. Yeah, that would be noise.
That's only 4 isolates. So even though it's a high percentage, it's pretty.
Yeah, exactly. And so in some of the cases we didn't. Yeah, exactly. So we're not analyzing 50 isolates there. We're just analyzing 4. Yeah.
Okay, a chicken's a bird, as far as I know. And the other slide had duck and geese separated. So is there an isolate just for avian population just versus mammal? That's what you're trying to distinguish between the two?
No, I mean, if you were to compare chicken, like the E. coli chicken feces, to that was found in let's say wood stork feces, you would probably get some different patterns, but you would probably get some overlap too. So there would definitely be an overlap, but and it would even depend on whether or not you used poultry from a commercial operation or whether you use backyard chickens. And so again, I think that the best analysis was by the gentleman who said was that noise, and I would say yeah, that was probably noise. Again, the take-home point is that most of these isolates are categorized as human, and the other take-home point is that this is a fantasy. So I'll extrapolate. Just to get the conversation started. Okay, so but we'll go on with that over time.
Did you
Yeah.
get any samples above where the blowout was?
Oh, so sorry. Basically, you're saying, did I get any sort of negative control samples? No, there was there wasn't any place where you could go and get a negative. Yeah, but it would have been a good thing. Yeah, that would have been a good thing to do. Okay, so basically then, this is right at the time that this happened, we're seeing, from both libraries, fecal coliform and fecal streptococcus, we're seeing good identification as human sources. Oh, I had an actual. Now, sorry, I thought I had another slide. When we went back later, so of course the drain field got repaired. We went back about 3 months later and sampled at these sites again, and almost all of the isolates were classified into, we had quite a number classified into the raccoon category, a lot classified into bird and dog. And so generally then we were seeing bird, dog, raccoon, and very low numbers in the human source category. So again, after the drain field was repaired and we went back, everything looked good. And these were libraries that had about a 72 to 74% correct classification rate, as I remember. That was the accuracy of the libraries if you combine sensitivity and specificity. And they had over 2,000 isolates in each library. So the libraries were pretty robust, at least for that area of Florida. So that gave us some good confidence in proceeding with some of the library dependent methods. But as time went on and we started, since Jorge Santo Domingo is not here, I will say the word ecology. We became more cognizant of the role that the ecology of these organisms plays in our ability to determine their source. And I'll show you some of the data on that subject in just a second. Now, the other caveat in terms of using antibiotic resistance data, and again, if you go back here, we were using antibiotic resistance analysis. The other caveat that we kept coming up with in talking to colleagues and reviewers was that, you know, the question of, well, how stable is antibiotic resistance in a population? You know, if you change the feed of a herd of cattle, you will, then you will change the complement of antibiotic resistant the profile of antibiotic resistance in that herd. Obviously, if you treat them with any sort of antibiotics, then you have a good chance of changing the population structure. And because antibiotic resistance is so subject to selective pressure, anytime animals are exposed to antibiotics, or indeed anytime their feed is even changed, you get real changes in their gut flora. So the question was, well, is antibiotic resistance really the best sort of identifier to use in creating a library? And so a lot of folks were exploring the idea of using genotyping methods rather than the phenotypic antibiotic resistance analysis, and still keeping the whole library dependent idea going, but using Let's see if we're back. We're back. So we started doing a lot of work on those sorts of methods. And here we're just showing a ribotyping gel here, and this is so-called box PCR gel here. So what we started finding as we did more and more, both of antibiotic resistance analysis and of genomic typing of E. coli and Enterococcus isolates, was we found that not all E. coli and not all Enterococcus are created equal. And we ran across a phenomenon that we call differential survival. Now remember that Don said that there was an issue with the fecal coliform to fecal streptococcus ratio because you had differential survival of these two different groups in the water. Well, it turns out that even if you look at one group like E. coli, and this is where the ecology is coming in, we have vastly different rates of survival in the water of different types. And so this is a bit of a complicated slide, but I'll try to walk you through it in some sort of a rational manner. The experiment that we did, we called it a mesocosm experiment. We basically were putting sewage and dog feces into our buckets that had sediment in them. So we were kind of trying to mimic their survival in a water body. And we typed these organisms. We did the genotypic and the phenotypic typing. I'm just going to show you the genotyping work that we did. And we followed their survival across several days. So here's the day 0 data, here's the day 1, day 2, day 3, and here's the week 2 data at the very end of the experiment, so kind of cutting to the chase here. And then these columns represent, each color slash pattern bar here represents the number of isolates of a particular genotype. So here, this is what we found in our initial wastewater that we put into the mesocosm. And you can see that we had a preponderance of this type here that's in the gray bar. And then we had several of this type here. We only had 1 of this type right here. We had 3 of this type right here, and so on. So the length of these pattern bars is proportional to the number of isolates. And so you can see that in the initial wastewater that we inoculated the mesocosms with, this pattern here dominated. And this is the initial waters. This is actually a Hillsborough River water. It's from one of our local rivers. And you can see the complexity of the patterns that we saw in the initial water. But look what happened over time. So we mixed basically wastewater with water. And then in the water column over time, we lost most of our original patterns and ended up with just a few dominant patterns that were not even seen in the original water or wastewater. So these were in such low proportions that in these particular starting material, for example, you see this pattern here coming up over here. But because they survived much better in this mesocosm, then they become the dominant ones over time. And so that just shows you that if you're isolating bacteria from a water body, depending on how long it's been since the contamination, you may get some representation of the original spill, but over time that population is going to dramatically change and alter because of this aspect of differential survival. And what you can see in the sediment, this is interesting. So here again, this is the same grouping as this here, because it's our initial inoculum, the wastewater. But in the sediment, you see that we have these 2 patterns here, or these 3 patterns here that became dominant over time. So what eventually became dominant in the water column was these initial patterns that were already present in the sediment that came from the Hillsborough River. So these were the survivor types. And over time, even after 3 days, they came to dominate the population in the water column. So what I'm getting at is that when you have this dramatic shift in population over a relatively short time frame, then the reliability of what you're trying to see that went in is going to be affected by their survival when you get these guys all tanking and dying at a really quick rate. So that was one major problem that we were seeing with our library-dependent methods. Here's another issue. So we're working with both antibiotic resistance analysis and ribotyping, and we found that depending on which method that you were using, you'd get really different diversity in the feces of the organism. So this is our genomic ribotyping patterns, and this is our phenotypic antibiotic resistance pattern. We are testing feces from human, cow, and horse. And so what you see is that the diversity in human feces by ribotyping is relatively low as it is by antibiotic resistance analysis. So this represents sampling efforts, the number of isolates that were typed, and this represents the number of unique isolates. And so the lower these curves are, that means the less diversity or the fewer unique types were in that sample. But what you see by the genomic patterns is, for example, we have really high diversity in the horse. And so even after we type 15 isolates from a horse sample, on average, we've looked at 9 different patterns. And so we would have to type a lot of isolates from horse feces to get some sort of a representation of diversity and adequately sample the population in the horse. And yet if we look at these cows here, the diversity is relatively low. But if we switch over to antibiotic resistance analysis, we see high diversity, really high diversity in both horse and cow. So the take-home point of this slide is that here, at this point, one of the pressing questions in library-dependent methods was, well, how many isolates do we type from each fecal type, and how many isolates do we type from each water sample? And the answer was starting to be, well, it depends on which method you use, and it depends on which animal feces you're talking about, and on and on and on. So it was starting to become really an issue of what sort of a rule can we make about how do we capture and where do we stop with the typing of these organisms? Have we done an adequate sampling effort? And if not, that's going to really affect our ability to assign isolates to the correct source categories. Here's another example. This paper was published in Water Research. And what you see here is these are box PCR types from a stormwater system. And the clear symbols represent samples when it wasn't raining. And so this water had been sitting around in the stormwater system for a while. And so you see that the diversity of E. coli is really low. We type 15 isolates and we only get 3 different distinct types. Enterococcus, somewhat low. We type 20 isolates and we've only got 6 different distinct types. Now these are going to be the survivors. They've been hanging around in the stormwater for a long time. But if we look at the Enterococcus and E. coli from raging stormwaters right after a rainfall, look at the diversity we see. We've typed 20 isolates and we're nowhere close to the diversity of E. coli and Enterococcus in that particular sample. So again, trying to establish a rule of, gee, how many isolates do we type in a sample? Well, I don't know. How long is it since it last rained? What's the temperature? Is it marine water or fresh water? And so you can see that we're starting to get into a lot of questions about how do we adequately sample the diversity. And unless you adequately sample the diversity, again, you really have no idea of whether or not your library is going to be accurate. So once again, Don Steckel is up to his evil ways, and so he and I were writing a review, and we were batting around the idea of, okay, so over time, from 1996, when the first library-dependent methods were published, up until 2005, how do we represent the success of the library-dependent methods? And we decided, one of the conundrums was, well, if you have 2 possible source categories, human and animal, then your chance of getting an isolate into the correct category just by chance is 50%. So if you have a 70% correct classification rate with a 2-category split, you're only 20% benefit over random. So we call this benefit over random. How much better can you do than random classification? Now let's say that you have 5 categories, you know, human, pig, dog, cow, horse, and you get, so where the random rate of correct classification will be 20%. Just by chance, you should get 20% right, if you have 5 categories. Well, then if you have a 50% correct classification rate on a 5-category library, now your benefit over random is 30%. You know, not 20%, where the 70% sounded so great when you had the human animal. And so we were batting this idea around of, well, how do we, with these, we're trying to compare these library-dependent methods over time. So again, we came up with this term benefit over random. How much better does the library do than random classification, than what you would expect from a random accuracy? And so interestingly, over time, you can see this definite trend, and I'm sure it has, I don't think we did the correlation, but I'm sure that it has a significant correlation coefficient. And you can see that this benefit over random is dropping over time, and this is as people were building bigger and bigger libraries, and they were using more and more source categories and finding more and more overlap promiscuous isolates. But the real kicker here is you see these white dots. These white dots were cases where blind isolates were used. And so if you had blinded source categories and external challenge, meaning isolates that came from outside of the library, then your correct classification rates dropped dramatically. A lot of this work was done establishing the correct classification rates of the library with isolates that were in the library. So using an internal sort of correct classification rate. And that's not right. You have to use external isolates that are outside the library to really establish whether or not your library has any accuracy in categorizing isolates that aren't part of it. So again, that was a review that we published in 2007. So overall then, and I'm going to run out of time pretty quick, but overall then, we discovered a lot of challenges over time with utilizing, developing and utilizing these library-dependent methods, including the size of the databases, the expense for making the databases, that you had to update the databases because they aren't, the population in a given watershed isn't stable over time. The larger the database, and Don touched on this, the more we tended to see these non-host specific or promiscuous patterns, making the data very, very hard to interpret and making this a fantasy. So the field in many ways started to segue over to library independent methods, and that was due to some of those challenges that we were facing in the library independent methods and library dependent methods. And please don't take me to be saying that I don't believe that there's any use to the library dependent methods, because I've worked with them a lot. I know a lot about when they're useful and when they're not. But the sort of our fantasy back in 2000, as Don mentioned, in the halcyon days when we thought everything was going to work, we're like, we're going to make a database and it's going to cover the whole United States, and we're going to know everything about where all this stuff is coming from, and maybe we can even do Europe too, and we had these grandiose visions, and they didn't quite work out. But library, and I'm not going to tell you that library independent methods are a panacea either. They have their own issues as well. But here then, the basis of this, as Don mentioned, is that we know that some microorganisms are confined to the GI tract and the feces of particular animal host species. And so finding a signature, some sort of a gene perhaps, to identify these microorganisms, if we can do that, then we can look for that signature in the water and try to determine the source of contamination, tracing that signature back to its source. And very frequently, the signature that we're looking for is, again, DNA from a particular gene that belongs to a particular organism that's associated with the feces of a particular animal, and we can use PCR to be able to detect or quantify that gene. I think I'm going to skip this slide. You'll have it in there, but this is basically just kind of a blueprint for how we would go about developing a microbial source tracking library independent method. But I do want to reiterate this point here. It's still extremely important to test the PCR method if you're looking at a PCR method for sensitivity, specificity, and the other performance characteristics that are so important. So yeah, this sounds simple. So you identify some sort of a gene, an organism, you find the gene, you develop an assay for it, and voila, you've got your library independent method. But we are talking about then looking at specificity, so collecting, feces and waste from a lot of we call these non-target host sources. So if you have a human marker, then we're looking for that in all of these different host species, in the feces of all these different host species, and hopefully we don't see it, because if we do see it, we don't have a very good marker. And I would say a ballpark that we like to see for specificity of a candidate marker would be around 90% or so. If it's much less than 90%, we aren't going to pay too much attention to it, because that sounds like there's way too much wiggle in that particular marker. Now, sensitivity is really important too. So we need to know that in our host population, we would like to see that this marker is widely distributed, because if we only have, let's say, 10 people that might be, so let's say we have a couple of families on septic systems next to a river, and when their septic systems are low, their drain fields are in the water table, and we're thinking, yeah, we think that this is an issue here. But if our marker is only found in 1 of 100 people, and we've only got 10 people potentially contributing to this contamination in the river, then we're not very likely to detect our marker, even if it is exquisitely host-specific. So we have to have, this marker needs to have, broad distribution in the target population, whether that target population is humans or whether you're looking at a dog marker or a bird marker, etc. So we need to know, with sensitivity, we're asking the question, if the contamination from a particular source is present, then how likely are we to be able to detect it? And it's interesting because you'll see in the source tracking literature that you can test sensitivity using a lot of different types of waste samples. For example, if you test the sensitivity of the frequently used human marker, the HF183 Bacteroides, if you test that sensitivity in sewage, you always find it in sewage, always, always. But if you test it in individual human fecal samples, you're looking at more like 10 to 20 to 30%, depending on what you test for. And so that goes back to the issue that Don was saying about make sure that you have your question framed very completely, so that you know what sort of testing you need to do in order to get good metrics for the marker.
Why would that be? Was it just the concentration in just one person?
No, it would be more because not everybody's carrying this particular kind of organism. So that even though it's pretty widespread in the human population, you always find it in sewage, because sewage is an amalgamation of poop from, literally thousands or 100,000s or millions of people. But if you just test one individual, they may or may not possess the marker. Just like if you tested a population of 50 people and you were looking for, or let's say 1,000 people, and you were looking for blood group AB, you'd certainly find a number of people that had the AB blood group. But if you only tested 10 people, you might very well not find anybody. Yeah. So, okay. So now going on with the theme of sounds simple, how hard could it be to validate these library independent methods? Well, we have to know something about the limit of detection. Again, Don also touched upon that. So I like to think of the limit of detection as a quantitative estimate of sensitivity. So how much of the marker is in sewage, and how much, really importantly, how much, oh, this doesn't show up very well. This is actually a sewage plume. You can kind of see the brown there. This is brown, and this is the water. So this is actually a sewage plume in Mexico. But so how much can we dilute the sewage or fecal sample and still be able to detect the marker? If we can only dilute it 1 to 10, for example, and we lose it, if we dilute it more than 1 to 10 and we lose it, then it's not going to be a very useful marker in a water quality study, right? Because most of the time we're diluted a lot more than 1 to 10 before we start trying to pick it up. If it's only 1 to 10, you could probably look over there and say, ooh, look at that, all that dog poo, ooh. So, then your eyes become your tool and not source tracking. So we need to be able to dilute those fecal samples, 1 to 1,000, 1 to 10,000, 1 to 100,000, to still be able to detect them. And then we come to the question of conventional PCR versus quantitative PCR. Now, for the first, well, I'd say 5, 6, 7 years of library independent source tracking, we use conventional PCR. And so there we get a plus-minus answer. Is the band there or is it not there? And as Don mentioned, we can use dilutions of the target and kind of get a ballpark idea for the relative quantities. But with quantitative PCR, we can actually use this instrumentation to get a much better estimate of the quantity of the target in the sample. I thought I had another slide. Okay, I did want to mention that quantitative PCR traces can be really difficult to interpret. And just if you see the standard curve on these things, so basically you have, with the quantitative PCR, it's just like a chemical assay where you have positive control material and you make dilutions of that positive control material, and then you determine the signal, and you can plot then the signal versus the amount of the target. The really counterintuitive thing about quantitative PCR is that the more target you have, the fewer cycles of PCR you have to do. And so we call, where the signal comes up, we call the cycle threshold on this trace. And so the cycle threshold here would be maybe 20 or so, and that might be 10 to the 6 copies of our target. So your cycle threshold will be low when your copy number is high, when your gene copy number is high. By the same token, if you have only 10 copies of your target, then it takes many, many, many cycles of the PCR to come up. And so when your copy number is low, when you have few of the target genes, then your cycle threshold number is high. So that gives you those backwards plots, which is kind of confusing. Just in case you ever have to try and interpret that qPCR data, it can be a little bit counterintuitive. Okay, sorry, I know I'm flipping around a little bit here, but that seemed to make more sense. So just to show you a success story from an MST study, this was done at a beach called Ben T. Davis Beach. And it's a very popular beach. It's right off the causeway here. And this beach had been having a lot of exceedances for fecal coliforms and enterococci. And so we used a couple of markers for human contamination. We used the Enterococcus faecium ESP gene, and we used HF183, which is a gene that it's a 16S rRNA target from a human-associated Bacteroides. And we sampled at an area where there's a lifeguard stand and porta potties. We sampled on the beach. We sampled at a major stormwater culvert. And we also sampled at this ditch here. And what we noticed was that when the municipality would clean out, there's a lot of porta potties right along here. When they would clean out the porta potties, it's a little hard to see, but these are porta potties right here. And they would just rinse those out right on the edge, and the water's right here. And you can see this material that would be just kind of, I know, you're like, are you kidding me? So they would dump the big stuff into the truck, but then they like hose the things down and bloop, and it would go. And here's the beach, and here's our sampling site. So you're like, really, you're doing that? But again, so we did find a high preponderance of positive samples for the ESP and the HF183. And again, this is another scenario where, maybe you could look at that and go, oh, that's a bad idea. But, we were there doing the study, we were able to demonstrate it, and they completely changed their procedures. They moved the porta potties, they had change in all their protocols. But the more thorny issue is here. Now we kept sampling this stormwater drain, and we kept getting HF183 and ESP, these human markers. So we poured dye in the toilet of crabby bills over here. We had them TV the line here. I mean, nothing. We could find no reason for these human markers to keep showing up in this stormwater. So finally, by good old detective work, our buddy Byron Bartlett of the Hillsborough Environmental Protection Council, he is dogged beyond belief. And he was stomping up and down this ditch, finding something, he's gonna find something, and blop, his foot goes right in a big puddle of ick. Because what happened was that the sewage line that ran along here had broken, and it had a slow leak. And so what was happening was the tide would come in, it'd go up, it'd pick up the sewage from here, shloop, slosh back around in this storm drain, and then, make its way over here, et cetera. And so if we hadn't been getting those consistent human signals from the stormwater system, we wouldn't have kept pursuing, where is it coming from? Where is it coming from? Where is it coming from? So this was a really good example of where these library independent methods proved very useful. And then, so of course they fixed this, sewage line, and then our lovely positive control beach became just like all other beaches. we had no more yuck yuck. I know, we had to find a new one. There was no beach as good as that one. That one was so good. Yeah, but I'm sure people who are swimming there are much healthier now. So I'm running out of time, but so we've done quantitative PCR studies, and this is a nice one that we did around the Hillsborough or the Tampa Bay area, and you can see here's the Gulf of Mexico, so we sampled in saline and in freshwater sites. Here's the Hillsborough River up at the Green Swamp. Now, one of the things about quantitative, or about PCR in general, is that you can have problems with something called inhibition. This is what we look like when we find inhibition, or we were trying to think about inhibition. Oh no, because that's when there's stuff like humic acids in the water samples that doesn't let the PCR reaction work, and you have to test for that. And so this just shows the example of testing for inhibition using an internal amplification control. And you can see that, and what we did was we compared remember I told you about that cycle threshold? That's the number of cycles that it takes for the trace to come up on qPCR. So we compared the cycle threshold that we got for in distilled water for this particular assay. This was the HF183 assay, to what we saw in all of these samples that were spiked with positive control. And you see that here, when you're getting these cycle thresholds that are much higher than what we saw in the control, that there we're saying, okay, so now there is definitely inhibition of the PCR. And you really have to test carefully for it, and you have to account for it in both quantitative PCR and in conventional PCR. One thing I do want to show you from a study that we did in Doheny and Avalon Beach, this was in conjunction with the Southern California Coastal Water Research Project. We were testing for indicator bacteria, and we also tested for human polyomaviruses. This is a human-specific virus. It's found in about 30% or so of individuals. And we were also testing for Bacteroides and M. smithii and adenoviruses. And what we found was that human polyomaviruses and HF183 were correlated significantly to detection of adenovirus in the Doheny Beach samples. So this paper is it should be accepted pretty soon in Applied Environmental Micro. We just have some minor revisions to do on it. So this ended up being a good example of where we can use qPCR and actually correlate it with a pathogen in certain types of water samples. So the moral of the story is, if you're going to do a MST study, whether it's library dependent or library independent, ask questions. Don't just accept that, okay, this is methodology that's worked out and it's just going to work off the bat. Make sure that you really are critical about it and you think about these metrics that I've been talking about. And with that, I may have time for a couple questions, and then I think we're going to look at posters, right?